So remember, DNA replication is when DNA makes a complete copy of itself. So if we look at the four main steps of this process, we're going to start with an enzyme called helicase. And remember, if something's got an ACE at the end, it must be an enzyme, and enzymes are proteins made of a series of amino acids. So our enzyme is going to attach to the DNA double helix. At this point, the DNA is not tightly coiled up as a chromosome. It's spread out so that the enzyme can actually get to the DNA molecule itself. The helicase is going to unzip the two sides of the DNA molecule. So it's going to break the nitrogen base bonds between the two sugar phosphate backbones. And so it's going to move up along this DNA molecule, splitting the two sides in half. Next, in step 3, we have the base pairing. So we're going to use a new enzyme, in this case DNA polymerase. And DNA polymerase is going to pick up individual nucleotides and match them with the exposed base pairs on the parent strand that are now open. And it's going to bond the bases together and then bond the sugar and the phosphates together. And so it's going to rebuild a new DNA strand. So here we've got a DNA polymerase rebuilding on this parent strand, and then you have a separate DNA polymerase building on the other strand. So you see here we're going to end up with two complete copies of DNA where we started out with one. ACE, that means again that this is an enzyme. Polymer, this is an enzyme that's going to build polymers of DNA. At the end, step 4, the enzymes continue for the entire length of the piece of DNA, and then they fall off once they've reached the end, and we end up with two complete copies where we started with one. So I want you to look at these and recognize that yes, they are in fact identical, and each copy has one side that was the parent strand and one that's the new strand, and this is called semi-conservative replication. I'll show you this video here. It doesn't have any sound to it, so I can just talk over what's happening. It's just another illustration of helicase and DNA polymerase doing their work. So we're going to start with a piece of DNA, and helicase is going to come in and unzip or break apart those bonds. Now we have two different DNA polymerases. They're going to move along, read the matching base pairs. They're going to bring in another one and bond the sugar phosphates together as well as the matching base pairs. And as we progress, notice that we end up with two identical strands of DNA. Each one, half is the parent strand and half is the new strand. Here we're calling it template and new strand.